It's a real pleasure to take part in this bilateral exercise and learning about each other's research. And I have to draw your attention that I will be moving away from just like Diccio Ripandelli yesterday, I'll be moving away from biomedical aspects and go back to a little bit to the plant and agriculture aspects. And I'll be describing you my research, an overview of my research this morning in the next half an hour. And I divide it in two main sections. One is signaling that takes place between the plant-associated bacteria and the plant. And the second is about the plant microbiome that I've just initiated, some research in that direction. So most of my research in the last 10 years has been involved in signaling, and the reason for this is that our world has really been revolutionized by the idea that bacteria do not want to live as solitary organisms, as we've been taught. They want to live as part of communities. So now we microbiologists accept that bacteria are always looking to form communities. The reason being that a community behavior usually tends to be a lot more powerful, a lot more resistant to attack by other pathogens and by the environment, by abiotic stresses like heat and salt and lack of water. So communities of bacteria are much more resistant and much more persistent in nature. And very importantly, they're much more virulent as well. Bacteria tend to attack when they are in large numbers, because by doing so they tend to defeat the host that they invade. Now, I don't want to talk about the research that we've done on a monospecies setup, because most of us microbiologists that have studied signaling that takes place through the production and detection of small signal molecules that I'll just now get into has been done on our favorite organism in the lab. But in fact now, the next step of signaling and the next challenge that we have ahead is, in fact, if we look at bacteria in the wild, in most cases bacteria live as interspecies communities. And, if we go to the soil, if we go to the gut, if we go to in our mouth, if we go to just about any niche in the world on Earth, we find that bacteria are part of multi-species communities of hundreds, if not thousands, of different bacterial species. And to make things even more complicated, in my studies, I look at plant-associated bacteria, so these very complex communities live next to the plant. So what is the plant doing? Are these communities communicating with the plant, or are they just independent? And obviously this is something that we need to look at, and that's what I'm looking at. So what I'll do now is I won't focus on the monospecies setup. I want to focus on the next stages of this challenging communication research, that's interspecies and interkingdom together with the plant. Now, if I just put this slide here, signaling languages in bacteria, it's still a new field, and this slide shows some of the signals, some of the languages that bacteria produce in order to communicate. Okay, and this is, in my view, is just the tip of the iceberg. We will find many more in the future. But what I want you to see is that these are small, very simple molecules, which can be produced, and usually they just diffuse in and out of cells. They don't need any active transport, which makes things very powerful. So you have a bacteria, you have a low density of population of a bacteria. There's a low density concentration of the molecule. As the concentration of the population increases, you have a high concentration of the molecule, and these systems of signaling are very, very simple. You have a signal generator, so a gene, a protein that makes the signal. And you have a signal responder, a protein that binds the signal and responds to the signal and this usually, direct interaction leads to regulation of gene expression. And also in this slide, which I've taken from a colleague, there tends to be, if we look at virulence, which we're very interested in, tends to be a harmless scenario, and this tends to be an aggressive scenario, okay? So, for example, an invading pathogen into a human or into a plant, the initial phases of growing from a low density to a high density is rather inconspicuous and hiding. When it comes to a high density, they start firing their guns, and by doing so, the plant can detect the presence and also respond to, but it could be too late because the bacteria are already in a large mass, okay? So what I want to show you here, just some examples, is my final really general introductory slide, is that a lot of research has been done in this direction, and what we can see here, there's a lot of very important bacteria that have been studied for signaling, which is called quorum sensing also, sensing a quorum, a concentration of cells. And what we can see is that a lot of the phenotypes that are regulated by this signaling, by this quorum sensing, are extremely important for virulence, or also beneficial bacteria use it to be beneficial and to make symbiosis with the host. What I'm proud to say that the fundamental basics, the textbook research has really been done on plant-associated bacteria. And then the human pathogens and the animal pathogens have followed. So I've initiated my work many, many years ago on bacteria that associate with rice. I used the rice model system for obvious reason. We all know the importance of rice worldwide. And I thought, which are the most important bacteria that associate with rice? And are these bacteria using signaling in order to be pathogenic or beneficial? As you can see here, there are pathogens, and there are also beneficial bacteria. Most bacteria that live beside plants are beneficial. In fact, they are very important as we come to in the end of my talk. They are very, very important for the health of the plant. So my question was many years ago is, these are the major bacteria that are interacting with rice. Are they signaling to each other? And I don't want to go through, obviously, this work, but the answer is yes, they do. They produce signals. They communicate with each other. And this communication in the forming of communities is very important for virulence or for the plant beneficial aspect. But what I want to do now, I want to focus what I think are the most interesting results that we got, the novel results that we got a little bit by chance. So we started looking at Xanthomonas oryzae, and which is the second most important pathogen of rice. This is a vascular pathogen, so it gets in through the leaves. Where the water gets in and infects the vascular system, and it blocks it. It's a very serious pathogen. The United States has in fact labeled it as a bioterroristic agent because we lose 10% of rice every year in Asia due to this pathogen, and if this goes up, we can create world famine. And the second class of bacteria are the Pseudomonas, the rhizosphere Pseudomonas. These are bacteria that live just attached to the roots of rice, and they're extremely beneficial. They provide a barrier, and they also communicate with the I'll show you, they communicate with the plant, and they exchange nutrients, and they help each other. So they kind of protect, they keep a barrier, and they keep pathogens away. So we started looking at signaling in these two guys. And what we were really, really baffled by initially is that, as I said, we have a signal generator for a signaling and a signal responder. What baffled us when we started this work is that these two bacteria, the Xanthomonas pathogens and the beneficial Pseudomonas, we could not find a signal generator, but we could find a signal responder. So it's like basically saying to people, I can listen to my surroundings, to signals, but I cannot produce them. Okay, which was rather novel at that time. So we thought, wait a minute, we have a protein inside of these bacteria that can listen to signals produced by neighbors, maybe by neighboring bacteria, or are these proteins responding maybe to a plant signal? So I want to give you a picture of what I'm doing. I don't want to get into too many technical details, but this slide is actually very interesting because it's 4, 8, 11 years of work. So this is 2 PhD students, Sarah, she's now in the States, and one who's also in the States, and Sujata is a postdoc that she's now in Singapore. So what they've worked with this system, with this protein, and what I can tell you now, in fact, in Pseudomonas we call it PSOR, in Xanthomonas we call it ORER. This is a signal receiver, and it's responding to signals produced by plants. So small molecular weight, diffusible plant signals enter these Pseudomonas or Xanthomonas. They are detected by these proteins, which are basically orthologs. They're the same protein. And then they switch on gene expression. For the Xanthomonas, very important for virulence. And for the Pseudomonas, very important for the symbiosis with the plant. For all these, we have done bacterial genetics, molecular biology work. We've done omics. We've done proteomics together with Mike. We've done microarray with companies, and we've got all the implanta data as well to show this. And we have several quite good publications on the subject. So this was new, and then we went to the data banks. As you know now, the genomes of bacteria is basically an explosion. I think we've sequenced 25 or so in my lab. Now, probably every day there's tens of genomes being deposited in data banks. And it's basically so cheap now to sequence a bacterial genome, and so quick. So now this is by far not updated, but if I throw in these proteins here, we find orthologs in many different bacteria. And what is really important and exciting. The orthologs that we found are in plant-associated bacteria. So this family of responders is not found in any bacteria that is not associated with plants. So bingo, we've hit a very important signaling circuit between plants and bacteria. So now what is the next step? We are together with, I'm now collaborating very closely with Pete Greenberg in Seattle, which is probably the most important scientist in the world in terms of signaling in bacteria. And I'm proud to say that my PhD student, Bruna Coutinho, just finished with me. She went to do a postdoc there. She just started now, and she's working on finding the signal. Obviously, his lab is very well equipped, and we're collaborating, and we're working on different strategies to looking for these plant signals, which in my view is a family of related compounds. So there's going to be a major language with lots of dialects, because maybe there's going to be small variations to the molecule, to the signal among the different bacteria that interact with different plants. And what's exciting also is that since we published this work, we were the first. There have been other scientists. There have been, Steve Lindau in Berkeley, and also Sina Rezobian. There's been Gonzalez in Texas, and also a Chinese group working with Xanthomonas campestris, cabbage pathogen, have worked with this ortholog because they've bumped into their work, and they've also shown that these guys are responding to plant signals, and they are regulating very important phenotypes. And in fact, we've published in 2013 two very prestigious reviews on the topic, in TIPS, a novel widespread interkingdom signaling circuit, and in annual reviews on a more global, they invited me because of this work, a more global review on chemical signaling between plants and plant pathogenic bacteria. Okay? So, so this is very exciting. We're working on it, and now we have a very solid collaboration with Pete Greenberg on trying to find these signals. Okay, so in the last part of my talk, what I want to, as I showed you the slide at the beginning, what I want to discuss is the interspecies, the big challenge we have to study. We've been taught by our university lecturers as when you come to the lab, you have to be sterile. You have to make sure there's no contamination in your culture. In fact, we now know that's a very, very wrong way of doing microbiology. So if you have contamination and you do your experiment, probably you get much more realistic results than being sterile. So how are we going to study interspecies signaling? How are we going to study bacteria that interact with each other? Again, by chance, I came across this disease because when I say by chance, is because people contact me and say, I want to study signaling in my favorite bacteria, my favorite pathogen. So a Moroccan student studying in Italy, Taha Hosni, he contacted me and said, I would like to come to your lab to study signaling because I'm studying this pathogen of olive. Okay? It's a Pseudomonas savastanus. It's a pathogen of olive. In fact, it lives on the surface of olive trees all the time. And it's a real tough, I have a word for it, but I don't want to use it because it's a bad word, but it's a really bad guy. It stays on the surface and it just waits there. It waits until there is a damage on the olive branches, especially the side branches, like frost or mechanical damage or a bird coming to a pecker, and as soon as there is an opening, this bacteria gets in and forms a tumor. Okay, there's no transfer of DNA to plant cells like you might think happens with Dagobacterium. This bacteria starts producing plant hormones and causes apoplasia, and it creates this also called olive knot, and which is colonized by the bacteria, and the bacteria it creates its own niche. Okay. But if you don't do anything, it's not really a serious problem. I mean, if you're a good farmer, you look at it, you cut off the branch and burn it, and that's it. But if you don't do that, then you might get problems, and you might even kill the plant. But I'm not interested in the disease. I want to, I'm interested in signaling and in communities. So Taha came to the lab, and the first thing we did is we showed we were happy. We detected the signal, producer and the signal responder, the signaling system. We made mutants, so we inactivated these two genes, the signal producer, signal responder. And here we infected 1-year olive trees. And we could see that after 60 days, you have olive knots in the wild type if you infect the wild type. So you make a little scar and you just put some bacteria. If you do that with the mutants that don't make the signal or don't respond to the signal, you don't have the olive knot anymore. Okay? And you can measure, we measured it here. This is all published. This is great. Okay, this is nice, but no big deal. I mean, there's lots of pathogens that have been shown that signaling is important for virulence. what really excited us is that Taha also said, when I come to your lab, I'm going to bring another bacteria, especially one that's called the Nervinia toledana, which has been, in fact, the species has been described in 2004 from a Spanish group. In fact, toledana comes from Toledo, the word, which is a Spanish city. And what is interesting about this Nervinia toledana is that a harmless guy is, it doesn't do any harm to the plant. We, in fact, think it's beneficial at some stages. And it lives inside or on the surface or inside plants. And it was classified as a novel species associated with Pseudomonas savastani, with the pathogen induced. So it lived inside the knot. So he said, can I bring this to? I said, fine, bring it to. And I started thinking in the lab, this is exciting because what we have here, we have a scenario where we have a pathogen that makes a niche, and we have a guy that is not pathogen but lives in the niche. Do we have a scenario of signaling, community mutualism? Do we have a community being formed here? So we started looking at this, and the first thing I can tell you is that what was immediately exciting is that this guy here makes the same signal as the pathogen. So they speak the same language, okay? So then we set up an experiment which really gratified us because we did the olive knot, as I showed you, we did the infections of the olives. And we saw when we infected the pathogen alone, it took 60 days. And after 60 days, this is the knot volume in cubic millimeters. And we can, as I showed you also the picture, we make a nice olive knot. But what was really important, let me press this because I've got this fancy. What makes it really exciting is that when we co-infect the pathogens with the Nervinia toledana, which is not a pathogen, the olive knot is much bigger. And trust me, in plant pathology, this is very significant. So we have a bigger knot, okay? That means that the disease, by having both, is much more aggressive. This second experiment, which is really this is the first very exciting result, but this is really almost, really made us open the bottles, is that if we infect, as I showed you, with a signal generator mutant, so the bacteria then doesn't make the signal, doesn't speak the language, we have a very small knot because we don't have quorum sensing, we don't produce the virulence factors, we're not able to attack the plant. We're not able to recognize each other, and so on and so forth. When we co-infect the signal generator mutant of the pathogen with a wild-type beneficial resident, we re-establish the olive knot. Now, as I said before, we know that the resident and the pathogen make the same signal. So what is happening here is that the resident is cross-feeding the signal, is speaking the language which is detected by the pathogen, and by doing so will then activate its virulence factors, so on and so forth. So this is what it's telling us, is that the two bacteria, this is a 60-day process, the two bacteria are quite happy to live together, they're stable, they don't kill each other, and they share the signals. They speak the same language, and they share it. I don't want to, okay. This is just to show you, this is just a confirmation. If we do the same experiment as here, but with the signal responder mutant, we don't have the same scenario because regardless that the resident makes the signal, we don't have the protein inside the pathogen to detect the signal. So we have no knot formation. I keep putting the names of the pictures of the people that do this work. This is Taha, the Moroccan PhD student. He's now in Bayer, working for Bayer Crop Sciences. And Julia, who's working with me, she's a staff member of ICGEB, which is helping me in this project and also many other projects ongoing in the lab. So then, we got excited by this, and we contacted my friend and colleague Caio in Malaga, in Spain, because I contacted him because he's probably the leader in the world to study pathology, the pathogenesis of this pathogen. And what is nice that he has a microplantlet system of olive that in a few days, a couple of weeks, you infect your pathogen and you've got your nice olive knot. And we infected so Daniel, I'll show you a picture. Daniel Pasos in my lab, he went to spend a couple of months in Spain, and he infected the microplantlets with the pathogen, and you can see that in 35 days you have a nice olive knot. And we labeled the pathogen with the GFP, and we could see nice, nice knot full of GFP. This is not new, and Caio has done a lot of work to show that the pathogen really colonizes the outside, the near surface area of the olive knot. When we infect with the Nervinia toledana, as I said, we have no pathology, and we have basically not much growth as well. We just see some red. Here we made them red, not much red cells. It gets exciting when we co-infect, because when we co-infect, the first thing we see is that the Nervinia toledana suddenly grows much more, and it co-localizes. Now, I don't want to, again, because of limited time, we've done confocal microscopy, we sliced, and we published quite a good paper in microbiology, which also they put it on the cover, some of our pictures, that showed that these two guys strictly co-localize. So the Erwinia tolitana, when we look with confocal microscopy, we never find it alone. We always find it very close, almost touching the Pseudomonas celestana. It's like I can grow in the olive knot, but I need to be close to you. But at the end of the day, the knot is bigger, so we both benefit. And in fact, we even counted cells. This is a very, very important experiment. If we look at the open cells, 60 days knot, we count the cells. Basically here, what I want to say that the number of pathogen is much higher. It's higher if there is the Erwinia tolitana resident. So the pathogen is happier if it has the Erwinia with him. And the Erwinia, the harmless, cannot grow without the pathogen. When there is the pathogen, these are the dark filled circles or triangles. There's a lot more Erwinia. So both of them grow much more when they're together. So basically, what we're finding out is actually quite logical. Bacteria have evolved out there for millions of years, not alone but together. Okay, so they've evolved as part of a community. So what myself and other scientists are looking into, apart from signal sharing, is now what I think is very important is metabolic sharing. So if we evolved together for millions of years, what is probably happening is that some of the metabolism that we do in that niche, we do it together. We don't do it alone. So what we started doing is also, when it comes to omics, we started doing phenotypic microarray. And what we can see here, if we do a phenotypic microarray, so we look at, utilization of carbon sources, nitrogen sources, sulfur sources, so on and so forth, in collaboration here with groups that have even computerized this analysis by looking at, hundreds of biolog plates. If we do the Pseudomonas and we do the Erwinia alone, we see certain metabolic activity. The metabolic activity is low to high. But when we put them together, what I'm saying is that we see an increase in metabolic activity, right? So again, I'm giving you, I'm going through, I'm going a little quick, just to give you a picture where I'm going. So there are compounds which I'm excited about because these compounds are in plants, and bacteria use them as food, as carbon, nitrogen, sulfur sources. And some of these compounds, I think, can be utilized well only when the two bacteria are together, which that's when the mutualism, the chemical complementarity takes place. Okay, so this is just a fancy slide that I want to show you that we also did the microbiome of the olive knots. We took 9 olive knots in the wild, and what I want to tell you is that there's not only Erwinia in the olive knot. What we now know that the olive knot, 50% of the bacterial load, so half of the bacteria inside the olive knot is the pathogen. The other half is lots of other bacteria. So you can see I've shown you with one bacteria, but man, is it complex. Okay? So the pathogen comes and makes the knot, and it takes in friends, and also in here there'll probably be cheaters. There'll probably be bacteria that don't contribute to the community but benefit from the community. Okay, so I also, because got excited with this, we wrote an opinion in Trends in Microbiome that I think that what also other scientists are looking at, that the pathogen is coming in an environment, in a human, in a plant, in an animal, in an insect. It's coming in, and it's finding bacteria there. We're full of bacteria. We have 3 kilos of bacteria inside us. It's finding these bacteria, and there could be interaction. These bacteria, like I've shown metabolic exchange in our case, chemical synergism, but there could also be other things that happen. So it's something we have to consider. The pathogen coming in, meeting up with the guys that are there, what's happening, Okay, so last 5 minutes, Lawrence, just 5 minutes, is work that we've just started because, Mauro, I agree, is pressurizing us to look also at applications, and I think this, as we get older, is also nice that we also move away a little bit from basic research and we look at possible applied aspects of our research. So I think I really, actually, I quite like this input from him. So we started looking at how we can also use our expertise that we developed all these years into something more applied. And something that is exploding now is probably, as you heard before, is the microbiome. We have the human microbiome, which is very important for our health. And what is lagging behind, but we're now catching up, but plants tend to, especially in the developed world, the research in plant-related research is not as well funded as biomedical research, which is not the case actually in less developed countries. But anyway, the plant microbiome we now feel that is extremely important for plant health. And in fact, the Obama administration, colleagues of mine have just put in a proposal to the Obama administration for the funding of $1 billion for the next 10 years, so $100 million a year for 10 years on plant microbiome-related research. And hopefully there'll be some money from international groups as well. So what happens is that the plant is surrounded by bacteria in the roots, rhizosphere bacteria, epiphytes on the surface, and endophytes inside. And we think if we have a very healthy microbiome, we can increase yield and we can keep pathogens away. And moreover, what is very interesting, and also I know that Saudi Arabia has a desert institute for desert agriculture, very important also to help the bacteria survive abiotic stresses like salinity and drought. So we need to work at this. We need to understand the plant microbiome. And what we started, obviously, it's an enormous field, so we have to give some direction in my lab. You know, we have limited resources. Where are we going to go? Okay, these are some of the mechanisms that are important, how the bacteria, plant growth promoting bacteria act. I don't want to go into. So what have I decided to put my money in bacteriology? Hopefully it's a good choice, time will tell. It's what we call endophytes. So these you know, several years ago we thought, oh, some bacteria live inside the plants in intercellular spaces. But now with the omics technologies, that we've been able to, you know, over 90% of bacteria we cannot culture. So it was difficult before the omics. Now with the omics technology, we're finding out, wow, there's a lot of bacteria just like us that live inside plants, which are called endophytes. So since they're not very well studied, and in terms of application, it's very exciting because you can apply an endophyte to a seed or to a plant, and they get into the cell. It could also be then transmitted to the next generation. So the beneficial community can be transmitted. So what we put our money on, and thanks to also a 3-year funding from a foundation here in Italy, is that he said, you know, we are the first rice producers in Europe, which is not a big deal when it comes to terms of how much we produce, you know, if we compare to India and China or Thailand or Vietnam, but still, we are an important rice producer. So we got money I convinced the foundation to fund us on, we're gonna isolate endophytes of Italian rice. And we did a very big project. It's now 3 years that it's going. We isolated endophytes from roots, from stems, and from leaves. I don't go into the details how we do that. We collect the samples and we sterilize them and check that they're sterile and then crush them and get the bacteria out. And we've done 54 samples. It's a lot of work, both on rice that is grown in dry conditions and wet conditions, and 3 different growth phases. So this is like the graph that shows the logic behind our sampling. Again, I'm going a little quick because I don't want to spend too much time on the technical details. So the first thing we did is metagenomics. Easy. You isolate DNA of these samples and you look at the complete photograph. You make a photograph of the complete microbiology. And we're getting some pictures of what's happening. You know what kind of bacteria are in roots that are dry, grown in dry conditions, the plants, or submerged, blah, blah, and so on. So we're getting these pictures. But I think in terms of application, what is more exciting is the fact that in all these samples, we grow the ones that we can grow. So we put them on plates. And we isolated 1,318 bacterial isolates. And trust me, that's a lot of work because each one has to be purified, saved at -80, and then classified, characterized. And here we're beginning to see some, you know, again, I'm giving you some names, but these are the genera. We're beginning to see that, you know, if we do general dry cultivation, submerged, we have some common and we have some specific. Okay? But this is the big work. This is the very big work. Out of these, the final aim, the final goal, hopefully, of our work is can we find some that we can then make products into? Can we find some that then we can give to the farmers as a product, to give to the plant in order to enrich the beneficial endophyte population? So you have to start. You can't do that with 1,318. We have to get to a small number. So I don't want to spend time on the logic behind it, but you know, we've done some. First start, we've done some classification, and then we had probably 689 different bacteria, we thought. Then we do some in vitro tests. So we test for production of plant hormones, we test for growth under limitation, we test for production of antimicrobial compounds. Phenotypes which we think are important to make a good bioinoculant, a bacteria that can be utilized as an additive to agriculture. And we went on, and also the final test is very important. Can we put them back into the plant? So can we put them in the roots, and do they go back in and colonize the plant. So in plant endophytism. So again, we did all this work. Actually, this is 3 years of work, hard work of Iris Bertani in my lab, funded by this foundation. And now, oops, where we are now after 3 years, we have 12 isolates, which we think are very good. They get into the plant, they colonize it well, and from our implanter work, the plant is a little heavier, it grows, but it looks healthier. So now what I'm trying to do is I've contacted the biotech companies, and I said, this is all I've done in 3 years. So if you, are we going to do something together? I mean, I go to them, and as we discussed also some discussions yesterday, I come to you with 3 years of work. So are you interested in taking this further? Can we do some field trials? And do any of these 12 isolates, are they, or in combination or as single, are they potential products, potential isolates to make a bioinoculant product? So now I'm discussing with one company, and you know, hopefully I can get to visit them in the next couple of weeks. They now go back to me and said to me, we're interested. We'll let you know if you can come and see us and discuss this. Hopefully, you know, hopefully this will happen. Maybe it's a mistake. I'm contacting the big companies. If this fails, I'm going to move down the ladder to smaller companies. So finally, I think I'm going to skip this because I talk too much. So finally, what I you know, this was a slide that, you know, with these strains we also do science. We do RNA-seq to see which genes are on off in plants, but I don't want to. So some of these 12 isolates, we sequence. In fact, we sequence all the genomes of the 12 isolates. We're also doing some science to understand how they colonize the plant and which genes are important. So finally, this is my last slide. In terms of translational science, we also initiated the strains for plants, which I think is could hopefully could be something important that we could do in ICGEB, because I believe in locality. I don't necessarily believe that the strains we isolated here in Italy are going to be used in India, in Vietnam, or in Thailand. So we potentially could reproduce this kind of screen in other growing areas, in rice or other crops, in order to identify beneficial bacteria. Let me tell you, this is an enormous field right now. The use of bioinoculants is going up by 10% a year because we realize we have to reduce the chemistry. So big companies like Monsanto, Novozyme, Bayer Crop Sciences are investing hundreds of millions of dollars to look for bioinoculants, biopesticides, biostimulants in order to reduce the use of fertilizers and pesticides. Okay. Last slide is that together with Mauro, we've been discussing, and we've been very happy of this bilateral exercise that we're doing. We think the Gulf region, ICGEB member countries in the Gulf regions, they should be a little bit more active in terms of ICGEB and ICGEB Gulf country collaboration. So since I'm very interested in this kind of topics, ICGEB Mauro agreed to fund a workshop in Kuwait City next year, where what we thought we would do is that we would, rather than choose a theme, we would choose rather, we would get together a group of scientists that are looking at research strategies to improve agricultural sustainability in the Gulf region. Obviously, there's a lot of challenges there because you have desert conditions, you have high salt, no water. So I've done some research. There's several PIs in United Arab Emirates, in Qatar, in Saudi Arabia. And we're going to get them together in Kuwait City. And together with ICGEB and the Kuwait Foundation for the Advancement of Science, we're going to get them together and make some discussions. Each PI is looking at strategies to improve agriculture in this very particular area of the world. Okay. So I've got a lot of collaborations that I need to have a lot of collaborations because a lot of these guys have techniques I don't have, or they have greenhouses I don't have, or they grow plants that I cannot grow. So I don't name them all, but it's really it's a nice part of my work that we have a lot of collaborators. And at the moment, I've mentioned some of the scientists that have done the work I've presented, and this is the current members of my group. There's Iris and Julia, who are been with me 15 years. They are like helped me run the lab, and now I have a Gauri, PhD student from India, postdoc from Serbia. I have a visiting PhD student from China, and we have also some visiting scientists coming up in the near future. This is the funding. Okay, thanks. Thank you.